Okay, so this is part two of the introduction information to lecture two, and it's maybe a little long. So as I said in the last video, if you already feel very comfortable with this material, if you can explain what a gene is in your own words, if you know what gene expression is referring to, and what genotype and phenotype mean, you may not need this one. So if you feel very comfortable already with the topic, don't worry about spending time doing this when you have other, perhaps more important things to do. But if you feel a bit rusty, or it's been like a really long time since you took the prerequisite courses, you may find this helpful. So we'll start just looking at these topics. So basically a gene is a piece of DNA. It's on a chromosome somewhere, and it encodes, or it codes for, these are encodes and codes for mean the same thing. And what encodes actually means is it provides instructions for the production of a protein or a functional RNA. So there are some RNAs that do certain tasks in the cell themselves without being translated into a protein. So when we're talking about gene expression, so you will see many, many examples through the semester of what we call changes in gene expression, which change the pathway the cell is on and change what types of cells it can become, what it ends up doing in the body. And so when we're talking about gene expression, we're specifically talking first about the transcription of the gene. With a couple of exceptions, all the cells in your body contain the same DNA. They have the same genes, the same alleles, and so on. But you have all these different kinds of cells, and that is because different genes are being transcribed in the different tissues. So there's no brain-specific proteins being produced in your muscle cells, for instance, or no muscle-specific cells in your nervous tissue. Right? So there's a set of proteins and genes that are produced in every cell because they are needed for making the cell function. And then there are specific ones that are different in different kinds of tissues. And it can be very broad, anterior versus posterior specific genes versus very precise bristle, the formation of a bristle versus the formation of the cuticle on a fly. So we're talking about the transcription of the gene to produce usually messenger RNA. As I said, some RNAs are themselves functional. We may touch on that a little bit later in the semester. And then they go through the process of translation to make a protein. And the phenotype is the physical effect of that protein. So if you look at this image, which I grabbed from Pinterest, these are 4 little baby rats, and they have different coat colors. This one is cinnamon. This one is agouti. You can't see them very well, but this is basically a rat that is black, like this mouse, but has an additional genetic allele that causes little speckles on the hairs. And so creates this more grayish kind of appearance. This is a kind of a yellowish color, and this one has the same allele that this one has with sort of little speckles on the hairs. And so you can see these 4 different colors. Cinnamon, agouti is the name for this one, mink, and black. And so these are a set of phenotypes, but each of the phenotypes is dictated by having a particular genotype, a particular set of alleles at that particular gene. We're going to go through a couple of examples of types of mutations, types of alleles in genes, and how we think of them dominant versus recessive and so on, which we'll go through in the class. But the key thing is to understand that the phenotype is the physical appearance, the physical effect of the protein, which could be, a phenotype could be having hemophilia. Your blood doesn't clot, right? Nobody can see that unless you get a paper cut and are bleeding profusely. So it's not just the physical features, but it's also the proteins in your body, the enzymes, whether they're doing their job or not, and so on. And the genotype is all the various alleles of the different genes that you have. Okay, that was a bit long. So this is the central dogma. So here's our gene. It's a segment of DNA on a chromosome. It goes through transcription, and some of the RNAs go off and do jobs. This one forms ribosomes. This one is part of the translation process. But most of them are messenger RNAs, and they go through that translation process and produce proteins. And we show the polypeptides. These are strings of amino acids, and we show them kind of coiled up like this because the protein has to assume its correct shape in order to do the job. And so if you make mutations in the DNA and you alter the DNA sequence, your RNA sequence is also altered because the DNA is being copied. And then if that mistake occurs and the wrong amino acid or amino acids are incorporated in your protein, it doesn't fold correctly, it doesn't do the job either at all or not very well. And so we see what we would call a mutant phenotype. Okay, so this is a rapid run through, but if this is very mysterious to you, you will need to review. And I do have some materials from my my introductory genetics course that I can let you look at if you feel that you need it. Okay, so if we were thinking about the gene, go back to the DNA, and we think about the gene. Gene contains three sort of general regions. The first is regulatory region, and there is a promoter and proximal control elements. Proximal means nearby, and so basically within a hundred or so nucleotides of where your transcription begins, you have three types of elements. You don't need to memorize these. It's just you need to know that there is a promoter region. Upstream of that, there are additional special enhancers and silencers and so on, which we'll talk about very briefly here, but you'll see examples as we go through the semester. We'll talk about sort of how you map those control elements and so on in some organisms. And then there's the transcription unit. This is the part that gets transcribed into messenger RNA. And then there's a terminator. We aren't really going to talk about it, but there's a specific sequences at the end of the gene that stop transcription from continuing, and it produces only the messenger RNA. And then I don't know that this is necessarily needed. I don't think we'll be covering it, but basically there are exons and introns. The exons form the mature message in eukaryotes. In eukaryotes, the introns are little spacer regions that are spliced out. They're removed before the transcript is completed. And they do allow for some flexibility. So one gene can sometimes produce multiple proteins depending on how you put together the exons. I kind of think that that's all you need to know about that for this course. Okay. So the transcription unit is the part that is actually being transcribed into messenger RNA. And so the messenger RNA is produced. The DNA is unwound a little bit, opened up a little bit, and your RNA polymerase is moving along in this direction. It's reading one of the strands and it's producing an RNA that looks like the strand above. No, it's actually reading the top one, isn't it? It's reading the top strand and producing an RNA that looks just like the bottom strand. If you look at the 5', 3', that will help you. If you need help with 5' and 3', just let me know. I can produce some additional information. And so the messenger RNA that is produced is going to have the coding information, and it's going to be translated. The ribosomes are going to read it, and they're going to read it 3 nucleotides at a time, and they're going to incorporate an amino acid that corresponds to that particular sequence. And I think the details of translation, I don't think you need to really have every factor and so on in your mind, but if you understand it at this level, I think it will help. Okay, now there are these upstream control elements, and they are the proximal control elements that we've already talked about, but there are further away ones that are called enhancers or silencer elements, and these are the ones that give rise to proximal control elements and your general promoter, your Tata box. These are found in all genes, but it is these upstream enhancer or silencer elements that give you tissue specific kinds of expression. So expression only in muscle cells, or only in nerve cells, or only in the nerves that produce dopamine, for example. It can get very, very specific. And so let's just have a look. Again, this is a lot of detail, but just so that you understand. So this is where your RNA polymerase binds at that promoter, and your all your genes have, as I said, the proximal control elements and the promoter. But there are additional upstream sequences, and they can sometimes be far away. They could be like 1,000 or 2,000 or 3,000 nucleotides away from where your transcription start is. And the way they work is, oops, sorry about that. The way they work is that there are proteins that specifically bind the enhancer, or it could be the silencer. And then those proteins also interact with the transcription factors that are found right on the promoter of the gene. So it is this part that actually does the transcription. But if this is an enhancer, then the enhancer activates, allows for the transcription to take place. But there are also silencers, and the silencers actually will interfere with, prevent the expression of the gene. So if we are in a muscle cell and this is a protein that's needed for brain development, then you're going to have silencers that keep this shut off, say in a liver cell or a muscle cell or whatever cell it is that doesn't really need this this protein to be produced. But if we are in, say, a liver cell, then the liver cells are going to have these enhancers, these proteins that bind here, that loop the DNA over a little bit, and that promote the expression of the gene. So hopefully that's clear. This is a lot of detail, but this is just to show you that it's way more complicated than what we're probably going to talk about in the class. And the key thing here is that they can enhance or silence different genes, meaning the transcription of those genes, and then that there are tissue-specific sets of these. So there's a set of enhancer proteins in muscle cells that turns on all the muscle-specific genes. There's a set of silencers, maybe in those muscle cells, that turn off a bunch of genes that are not needed in muscle, like nervous specific, or digestive system specific, or something like that. Stuff that's needed only in the skin, for example, that kind of thing. Okay, and so the difference between having high or low levels of transcription depends on whether the specific factors are present. So for the core promoter, the RNA polymerase and some of the other factors are bound in both cases. But in a liver cell that requires lots of albumin to be produced, you've got the specific enhancers that will bind to the DNA and promote the expression of that gene. Whereas in a brain cell that doesn't need albumin, you might get a teeny bit of transcription there, but you don't have the right enhancers like these two here. These proteins, they're not found, they're not being expressed in the brain cell, and so therefore they're not binding to this sequence that is upstream, and they're not promoting the expression, so you get very little or no expression of the gene in that specific tissue. Okay, I hope that was helpful. It was a bit rushed. I'm mindful of not taking up too much of your time. But if you need more information or clarification, do please just ask, and we'll help you.